The agenda will be is listed here briefly. It's a quick microRNA intro, what we know about microRNAs, a little bit about the history, then highlight just a few recent studies. We actually will list a couple of them that you can then click from the PDF file that we sent directly, so that you can directly download these studies. Then in current methods, I will focus mostly on next-gen sequencing and microarrays, because these are still the most common profiling methods. And qPCR is kind of better known, and more people are exposed to that. And then in the last section, I will just highlight a few case studies of publications that I found really interesting in the field that were published within the last 2 years. And as Chris mentioned, of course, whenever you have questions, feel free to type them in. He will probably, Chris and Jason will answer them via the chat box, either directly to you or to everybody in the audience. If you're new to the microRNA field, you might not be you might not have heard of us because we're a very microRNA-centric company. Our headquarters is here in Houston. We have also a rather large office in China, and representation also in other parts of the world, like India, Korea, Japan. But fact is that we have customers in more than 40 different countries. So all the samples end up here in Houston. This is where our lab is. So all the arrays and sequencing is performed here. We entered the microRNA field in 2004, 2005. We were the first microarray service provider. And what the core technology is that we built the company upon is our microfluidic chip technology. So these are not your regular spotted microarrays, but these are microfluidic chips. And the special thing about them is basically that we can highly customize them. We never pre-manufacture any arrays. The arrays are always synthesized to order. And of course that allows us to keep up with the rapidly evolving field of microRNA research. This I mentioned already. We have customers in more than 40 different countries. This is just a rough representation, mostly in the academic field, but increasingly also from biotechnology and pharmaceutical companies, as well as government agencies like Food and Drug Administration here in the US, of course. We were also pretty strong besides the cancer field and the cardiovascular field, neuroscience field, of course. And because we can customize the chips, we offer a lot of arrays for species that are important in the agricultural field. So that's another strong point of ours. The technology, like I said, is a microfluidic chip. That's how the company started out with. In the meantime, we added other microRNA-related services like sequencing or validation using qPCR. And it was basically the microRNA arrays were the first application for this technology. The founders, when LC Science was founded in 2004, 2005, were looking for, they developed this technology and were looking for good application, and where you can take advantage of the custom features of the technology. And fortunately, they were visionary enough to jump into the microRNA field, which was really just starting out back then. Besides that, of course, we can synthesize also peptides on the chip, so we are now also focusing on the proteomics part and applications that you can use. Okay. microRNAs, what's the current status? What do we know? Typically, they're, of course, small non-coding RNAs, usually consisting of 20 to 22 nucleotides, which is true for mammalians. For plants, they're typically a little bit longer, up to 22 nucleotides. But the exact length, of course, and the length distribution depends on the developmental stage oftentimes. They exist initially as precursors, have a well-defined stem loop region as well as a hairpin structure, and the structure is very, very stable. Many microRNAs are evolutionary conserved. So for example, you will find that a lot of microRNAs are conserved between C. elegans and human. And the same, of course, is also true for the plant field. And what is clearly known is that microRNAs act as negative regulators of translation, and they do that by binding to complementary, more or less complementary, I should say, messenger RNA molecules. And by binding, they inhibit the translation. So when they're expressed, they're typically expressed in a very high copy number. And expression is tissue, but not only tissue, also developmental and disease-specific. So that, of course, opens up the opportunity to use them as biomarkers and for diagnostic purposes. As of today, we have 21,643 known mature microRNAs, and this is gathered from 168 different plant, animal, and virus species. And all the currently known and experimentally verified mature microRNA sequences, as well as precursor sequences, are deposited in the public database called miRBase. And the number behind it, miRBase 18 in this case, always, of course, indicates the version number. The latest version is version 18. The latest update was in November 2011. And from what I know is that next update to miRBase 19 should happen pretty much any week now. The mechanism of microRNAs is rather complex. It's complex because each microRNA may control a couple of genes and not just one. And also the other way, it's true that each gene can be regulated by a couple of different microRNAs. Another thing is that initially, the initial belief was that one microRNA will target only one gene and will act on its own pretty much. In the meantime, it becomes more and more clear that there's always not just one particular signature microRNA responsible, but rather a signature profile. So a couple of microRNAs, typically around 15, that act together. It's estimated for the human species that roughly 1/3 of all human genes are regulated by microRNAs. And I'm sure that will increase as our knowledge expands. There are typically two different ways of mechanism. One is the so-called complete pairing. Basically, the microRNA and the messenger RNA are an exact match, an exact complementary, and that's predominantly in plants the case. So the microRNA binds tightly to the messenger RNA and triggers its degradation. In the human body, what is more prevalent is the imperfect pairing, so mismatches are allowed, which of course makes it so complex. So the binding is strong enough to repress translation, but not as strong as it would trigger the degradation of the messenger RNA. So it's pretty much a typically reversible repression of the translation. Just to give you an idea about the timeline of this field and to show how fast evolving it is, I won't go into the detail here of the small font sizes here. That's just so that you can read it up in the PDF file if you choose to do so. So 1993, the first microRNA ever was discovered it was called Lin4, and it was in C. elegans by Victor Ambros' lab, and a lot of people didn't really pay attention and probably even dismissed it because it was also a central challenge to the dogma of biology, which stated, of course, that DNA gets transcribed into RNA, and then RNA gets translated into the protein. And the existence of a small RNA molecule that actually regulates the DNA transcription, that was really not in the plan here. So then 2000, the first human microRNA was discovered and described. It was called Let7. And 2001, a couple of labs, Bartel's lab, Tuschl, and Ambrose lab, discovered a large class of these small regulatory RNAs, and that's when they decided to name them microRNAs. So since 2001 only, just 11 years ago, these microRNAs were officially named. In 2002 then, basically the identification of DROSHA, which is an enzyme, was the final puzzle piece in the microRNA biogenesis pathway. And I will show you the pathway in a little bit, how microRNAs are processed. But since 2002, basically, we know how they are, at least the outline, how they are processed. This was also the same time, 2002, when the Croce lab first reported 2 microRNAs responsible or involved in cancer, microRNA 15 and 16. So as the number indicates, it's a pretty low number for microRNAs 15, 16. These were one of the first ones that were discovered, and immediately the role in cancer became apparent. Then in 2003, the first experiments in vivo with silencing of microRNAs with artificial oligonucleotides were done. These are called so-called antagomeres. And yeah, as the field started to evolve, the methods went along with it and were developed with it. So in 2004, it was the first time that microarrays were used to profile microRNA expression. And this is basically the time when, end of 2004, early 2005, when LC Sciences was founded and entered the field. 2005, then there were also 3 major studies about the link between microRNAs and cancer development. They are listed here, so you can then look at them by clicking on the link from the PDF file. In 2005, also, the next-gen sequencing became more available and was used for the first time for small RNA discovery and analysis. So again, it's just 7 years ago that this was done. In 2006, the miRBase, the public database that I mentioned already, went online. Back then at the Sanger Institute, and I believe right now it's at the university, hosted at the University of Manchester. This is the to-go place for experimentally verified microRNAs, as I mentioned, and basically the main source for sequences. They also deal with the have a blog there that keeps up with the current nomenclature, which is highly recommended. Then in 2008, microRNAs were detected in body fluids, and there's of course a whole range of them, but it started out with serum and plasma. In the meantime, it also includes urine. So basically, of course, it has a major advantage that you don't have to use any tissue samples any longer. It's much less invasive. This is a brief overview here about the microRNA processing. You basically start out, the first transcribed molecule is the so-called prime microRNA. It's the primary transcript. And it has I'm not sure whether you can see my cursor, but you have this stem loop structure here, which can also, this bulge here indicates that mismatches are allowed, so these are not perfect complementary strands. And then you have the hairpin structure here on the right. And the left shows you that you have still a 5' cap region as well as a poly A tail. So this prime microRNA then gets processed by an enzyme complex called DROSHA and PASHA. What's happening here is that the 5' cap and the poly A tail at the 3' end get cleaved off, and you have the so-called pre-microRNA or precursor microRNA. At this point, the precursor gets then transferred into the cytosol and further modified by a so-called dicer molecule. And what's happening here is basically that the remaining ends at the 3' and 5' end get cleaved off as well as the hairpin structure, and you're left with a antisense microRNA duplex, which is shown here. And this star here at the microRNA just indicated is a remainder of the old nomenclature. With the latest miRBase update, this was changed. So basically, since you have two mature molecules of microRNA here, the one that was first discovered was just called microRNA, let's say number 1, and the antisense to that, which was typically discovered later, because it's oftentimes lower abundant, because it's degraded fast, it was given the star tag. Because it was never clear which one was which, the nomenclature was just recently changed to -3p and -5p. And again, I refer there to the miRBase blog, which covers that in detail. So these two mature microRNA molecules dissociate, and you have basically two potentially active mature microRNAs. Initially, it was thought that only one of them is active, but recent studies show that both can be functional. And yeah, these three mature microRNAs can then bind to the messenger RNA and inhibit translation. This is showing you the rapid increase in knowledge about the microRNA sequences. It's basically a time span here of just 6 years. It's from April 2005 to November of last year. And what is shown is the total number of mature microRNAs in the database, in miRBase. And like I said, you have miRBase exists in different version numbers, and so it's done out here at version 6 to version 18. We just cut off the initial versions here. And when you look at the number of publications about the microRNA field, you see the same almost exponential trend here. These are the publications listed in PubMed. From 2001, basically non-existent, to 2012 with a huge volume. And if you look at the microRNA papers that deal with that cover microRNA and cancer, you see pretty much the same increase. And not so interesting probably for you guys in Europe, but here in the US, the NIH just announced again another project where they are funding specifically projects that look at microRNA as cancer markers, and not just microRNAs, but other short non-coding RNAs as well. But I thought that was interesting, so they just announced this. But let's skip this. So why would you want to study microRNA in cancer? Of course, it's as pointed out, it's pretty much involved in assumed that it's involved in at least 1/3 of the regulation of 1/3 of the human genes. So obviously it's really critical. But specifically when looking at cancer, it's interesting because they can both act as, on one hand, oncogenes as well as tumor suppressors. Then they also have a role in cancer stem cell biology. People typically look there as at the effects of microRNA expression on metastasis and drug resistance. And just in general, microRNA is regulation or differential expression is crucial part of the tumor formation, the maintenance, and its metastasis. And I mentioned that microRNAs can act on one hand as oncogenes and on the other hand as tumor suppressors. And two examples are listed here. microRNA 21 is one that pretty much shows it's a guarantee that it shows up in differential profiling of any type of cancer. And it is an example of an oncogenic mir, or short form is oncomir. It's highly overexpressed in most cancers, like breast cancer, colorectal cancer, lung cancer, and so on. And so it's something that clearly can be used also as a diagnostic tool. It's not very specific though. It's because it covers such a wide. It's affected by such a wide range of cancer. And on the other hand, miR15 and 16 are examples of tumor suppressors. They target the oncogene BCL2, which again encodes a protein that's involved in cell survival. So when they're downregulated, of course, this protein that helps with cell survival is not expressed. But when you reinstate then expression of miR15 and 16, you can turn on that protein expression again and increase the chance of cell survival. When you look at the clinical potential of microRNAs, of course, the first thing that comes to mind is based on also what I covered so far is microRNAs as a diagnostic tool or as biomarkers. Basically, you use microRNAs signature profiles to identify certain. For example, you can differentiate between different types of subtypes of lung cancer or kidney cancer or things like that. Basically, you use them as diagnostic tool to improve the diagnosis that is oftentimes a challenge for the clinician. Then biomarkers were found, microRNA biomarkers were found in bodily fluids like mentioned serum, plasma, urine nowadays, even breast milk. Then they allow, as mentioned, a detailed and very accurate clinical diagnosis, and with that it can affect or improve the choice of treatment and significantly improve survival rates. What can be used also for is to monitor and also to access to gauge the carcinogenic effects of environmental factors and toxins. Currently, there's a diagnostic kit on the market that's available for the clinician. It's called MirView, and this basically differentiates between the 4 main types of lung cancer, and it's produced by Rosetta Genomics. Then another field are, of course, when you move from the just purely diagnostical field into the therapeutic field, microRNAs can be used as tumor repressor drugs or basis thereof, or on the other hand, they can be used as direct drug targets as well. And they can be also used, and I will show a brief paper about that later, to understand the chemo and radio resistance. This is just, as mentioned earlier, a list of different reviews, very recent reviews, all from pretty much over the last 2 years, covering the different types of cancers. And as you see, it really covers the entire range that there is. So this just for you as a tool that you can read up on later if you like. So a very recent review that was published just recently this year is dealing with microRNAs in cancer management. It's basically covering both the non-microRNAs and their roles, and just summarizes the current state of what we know in the field. And it covers what is shown here in the graphic, basically both sides. On one hand, the oncogenic microRNAs like microRNA-21 that are highly upregulated in cancer, and then those that microRNAs like microRNA-15 and 16 that can work as tumor suppressors. Then also a paper that was just published in May of this year. It's not really specific for cancer, but it's an excellent paper especially if you're new to the field. It covers basically in detail the different profiling methods that are out there, and of course it focuses on the 3 main ones: qPCR, microarrays, and next-gen sequencing. And what I particularly liked is this little flow chart here on the right, where it gives you handy tool to decide which method makes the most sense and is the most cost effective. Because something that we see over and over again is that customers come to us and they tend to want to use the latest and coolest technology, meaning next-gen sequencing, but maybe the goal of the experiment doesn't really justify the cost, or maybe this particular method, in this case sequencing, doesn't even make sense to answer the question that is being asked. So I really like this, and this is a really good overview article. Actually, the only drawback is that they misspelled our company name. We listed as LC Biosciences instead of LC Sciences, but that's the only flaw that I could find. This is a list of microRNA research tools, the different methods based on the microRNA pretty much workflow, going all the way from identification, basically discovery, to then profiling, finding out the targets, the pathway analysis, and functional analysis. So when we start out with the first one, the microRNA identification, genetic screening, and then direct cloning and sequencing was, of course, the initial way to go. And typically these sequences then were validated using either qPCR, custom qPCR methods, or what we did a lot were in the initial stages were tiling microarrays. Of course, then computational methods where you try to predict microRNAs. There's a whole range of them, and we will include an extra slide with all kinds of computational tools here. And then, of course, more recently, then since 2005, next-gen sequencing, and this clearly has given the field a big push. So the rapid increase in known mature microRNA sequences, as shown earlier in the graphics, is clearly due to the next-gen sequencing success. Then when it comes to profiling, initially northern blot was used, and of course you can use in situ hybridization and real-time PCR. And then also microarray profiling, as well as some people use next-gen sequencing for profiling as well. Then target determination, there is a whole array of databases out there. Target Scan, and just miRBase has also its own target database. And one side that I want to point out, because most people that working here on the cancer field are typically working with a well-characterized species like human or mouse or rat, is the database or data. The website is called mirfocus.org. It includes all the latest human microRNAs, and basically it's pretty straightforward. You enter the microRNAs that you found that are differentially regulated, and it gives you all the information that it can find about the potential targets and pathways. Then, of course, you can look at when trying to find out the actual target, you can look at the proteome expression analysis and gene expression, run gene expression arrays, ideally, of course, of the same samples. You can do pull-down assays, 5-race analysis, and more and more upcoming is degadome sequencing. And actually, this is something that we just added recently to our service as well. Then, yeah, for the pathway analysis, there are different bioinformatics tools. In the meantime, even commercial ones. But that's pretty much, yeah, it's still a field that could see some significant improvement, hopefully, over the next years. Then when it comes to functional analysis, there are lucifers assays, of course, knockdown experiments that you can perform. Then antagomers, as I mentioned, you can use for microRNA inhibition, and you can do also microRNA mimicry experiments to do that. From all of these methods, I will, as mentioned earlier, focus on next-gen sequencing for the discovery part, and then for the profiling, I will focus on the microarray part. So there are typically three steps involved. The discovery is done initially, like as mentioned earlier, with sequencing and so on. And for the profiling, typically microarrays were used, and then for the quantitation or validation, qPCR. And then with next-gen sequencing, when that became available, that was of course the really powerful tool for the discovery step. And nowadays, our workflow for less well-characterized species is typically we do the discovery with the next-gen sequencing, then the profiling for a number of reasons, then based on custom microRNA arrays, where we develop the chip layout based on the sequencing results, and then validation with qPCR. But since we're talking here about well-characterized species like typically human, mouse, and rat here in this audience, I will focus on just the individual applications. No matter which method you pick, you have to make sure that you have a good sample. So there's a whole range of different commercial kits, as well as, of course, the good old-fashioned Trizol method is out there. No matter which method or which kit you pick, the most important thing is that you make sure, which sounds kind of obvious, but it's really not, is that you make sure that the protocol retains the small RNA. There are a lot of Trizol protocols as well as commercial kits out there that call themselves total RNA extraction methods, but they are not really, because they were developed before microRNAs were known. So they fail to retain the smaller RNAs. So if you're familiar with Trizol, you will probably get very good results if you modify the protocol. And we have a modified protocol on our website under frequently asked questions. And if you're new to the field and not experienced with Trizol, we would highly recommend to use a commercial kit, simply because our observation is that people really struggle to get the Trizol method working right when they're inexperienced with it. And here's a list of different commercial vendors and 3 very good kits that we have great experience with that our customers use. Non-Gen Biotech offers a wide range of specialized total RNA extraction kits. Then QIAGEN has a kit which is called miRNA. And when you decide to go with a kit from QIAGEN, make sure that you're picking this one and not the one with a very similar name, but it's called, it has a shorter name, it's just called RNA kit. That is an example of a kit that does not retain the smaller microRNAs. And then Ambion, the miRNA kit, gives you also really excellent results. Basically, no matter which method you pick one that's specific to your sample type, of course. There are specific kits for, let's say, FFPE samples, or when you're dealing with urine or blood samples, there's a different, typically a different protocol as well. And so pick one that retains the small RNA, pick one that's good for your sample type, and no matter which one you end up picking, stick to the same kit throughout the entire experiment. While they all work, they will all give you a slightly different profile. So if you switch kits during the experiment, there you're introducing a variation that you might want to avoid. Then to QC your sample to get an idea about the quality, you can look at the UV spectrum. So we typically use the nanodrop method to look at the ratio of 260 to 230, which should be above 1. Then the 260 to 280 ratio should be above 1.8. We also sometimes run bioanalyzer gels. There we typically look at a RIN value of 7. But this, of course, all this only applies to regular tissue samples or cell line samples. If you have blood or serum or urine samples, all these criteria kind of void, and basically it's pretty much trial and error at this point in time. The bioanalyzer we don't run routinely simply because it doesn't really give you a good indication about the microRNA quality, but you get a good feel for the overall sample quality. So if in doubt, we do run it. Otherwise, we just run the array and see how the sample looks. If you, of course, the best method, but also the slowest, so really low throughput method is to run agarose gels, as shown here, for example, or urea page gels. These are samples of good extraction methods versus poor ones, or degraded samples. These gels were run with different types of extraction kits. So on the left, you see always is here at the bottom is the microRNA portion very well retained and conserved and well defined, whereas on the right hand it's not retained, either because it's completely degraded or because the extraction method was not right for this approach. Then a big misconception seems to be that when it comes to biological repeats and the number of repeats that you need, the misconception that I was referring to is simply that some people think that only because you're using the latest technology, meaning next-gen sequencing as a method, that for some reason you don't need any biological repeats any longer. So of course that's not true, because the fundamentals of statistics don't change. So instead of running one control and one cancer sample using next-gen sequencing, which gives you two results which are statistically completely meaningless, we would always recommend to for the same price, run, let's say, 3 control and 3 treated samples using an array or using qPCR. So the number of biological repeats really is independent from the method. The number that we recommend, it depends on the sample type. If you have a typical cell line or tissue samples, we would recommend a minimum of 3. That's certainly a good number just to get a feel for if differential expression is happening or present or not. If you switch then to patient samples, of course, you're in a completely different ballpark. There you're looking at a minimum of 10 per group. It really depends here on how far you can narrow down your patient population based on age, disease stage, sex, race, and things like that. So anywhere between 10 and 30 samples per group is appropriate there. Another point is that you might want to make sure that ideally the number of samples per group is equal. So it doesn't make much sense to have, let's say, 10 treated samples versus just 2 controls. Ideally, you have 10-10 or 5-5, something like that. As mentioned, I will focus here on the arrays versus sequencing and the main advantages here. Microarrays are, of course, proven over decades now. They had initially a pretty bad reputation due to the fact that some of especially the earlier spotted glass arrays had really pretty horrible spot shape. But that has evolved, and with our microfluidic array, that's not an issue anyway. Then it's a very high throughput method. It's streamlined. It can be automated. The analysis is pretty much straightforward. The data that you get are easy to understand, and the turnaround time can be very short if you do it yourself, of course, anyway. But if you use a service, it's anywhere between 5 days, business days in our case, to typically 2 to 3 weeks. And the cost compared to sequencing is significantly lower still. The advantages of rna-seq or next-gen sequencing, on the other hand, of course, is that first of all, you don't need to know the sequences that you're looking for. So whereas the microarray will limit you to known or predicted microRNA probes that you put on the chip, with next-gen sequencing, of course, you don't have that limitation. And you can also look at a whole other range of transcripts at the same time. You can look at messenger RNA, non-coding RNA, and things like that. It has also an increased dynamic range, and you can tune the sensitivity, which you can't really do with the other with arrays. And if you're interested in for example, single nucleotide mutations, this is clearly the way to go. Or if you're interested in isoforms of the microRNAs, which are a little shorter or longer or show mutations, then there's really no other way, pretty much, to do this. Also, it's a really digital solution, so meaning it gives you an absolute abundance, not just a relative abundance like arrays do. So this is just briefly our microfluidic chip. We have different versions, but this is our smallest one that we started out with in the microRNA field. You have 4,000 features here, and what is shown here is the electron microscopic picture. You have the liquid is coming in through these channels here and going into the reaction chamber, in which it faces then the sample faces the microRNA probe. And then it goes out on the other side of the reaction chamber into the channel here again. It's a closed system, so you have a silica base here in which we etch these reaction chambers, and then it's covered with glass. So you have only one hole over here and one on the other side, which is covered here by the thumb in the picture. That is the inlet and the outlet. Other than that, it's a closed system, and that has the advantage that, for example, you don't have to worry about things like dye oxidation, dye bleaching, or anything like that. Because we custom synthesize each chip using photosynthesis, we can keep up with a rapidly evolving miR-based content, so that's what we call miR-based synchronicity. So this is showing you again the number of, or the increase of microRNAs in miR-base, and currently we offer version 18. So basically, the fact that we're using photosynthesis and that the fact that we don't pre-manufacture allows us really to keep up with the latest updates. So if version 19 will get released tomorrow, then the next day version 19 will be available on our chips. There are two main incision points here that I want to highlight. One is the transition from miR-base version 10 to 10.1. Doesn't look like a big change here in the number of sequences, but it was a major change in the naming of the microRNAs. A lot of names were changed, as well as sequences were corrected. So microRNA number 1 before version 10.1 and after 10.1 might not have the same sequence. So that's something to watch out for when you're looking at older arrays or also primer data, qPCR data. And then the other big change, and that's still actually a work in progress, was with version 18 in November of last year. As mentioned before, the nomenclature was changed, and this was done so far for implemented for the human, mouse, and C. elegans. And of course the other species will follow, so that's why it's kind of a transition state right now. But this can really severely affect your analysis if you try to compare the different versions. This just briefly the workflow when we receive samples. Of course, we do first sample QC, sample prep, do then the actual hype, scan the chip, do the data extraction, processing, and do the data analysis and send you back the raw data as well as analyzed data. This is how a typical experiment looks like. You have a number of biological repeats. Then we run the arrays, single color arrays in our case, and then the initial heat map looks typically even worse than this. So basically, you see the whole range of up and down regulation, and it looks like a big mess. And it's always amazing to see how applying a couple of statistical methods just gives you suddenly a very clear picture at the end. So this is shown here. This is the overview heat map, and then we ran a t-test between the control and the treated group, and then the heat maps here on the right show you the up and down regulation based on different p-value cutoffs. And the p-value, of course, stands for the statistical significance. So the lower the number, the higher the significance. And if you plot these data, the control versus treated here, one group on the x-axis, the other one on the y-axis, then you get something that looks like this. And you see these clusters, and what these are basically the probe repeats for each microRNA. So for we have 4,000 features on the chip, and especially initially, we had a very high number of repeats. For example, I remember Drosophila had something like 19 repeats. So basically, we had 19 identical probes on the chip for one particular microRNA. And this is what is shown here. These repeats that cluster nicely together, meaning that the data are in very, very good agreement. Nowadays, we have typically 3 repeats. That's typically the average. For some species, of course, there's still way more repeats than that. And this is just briefly, again, you get raw data, layout file, and analyze data. I'll skip this. So then, as mentioned, the advantages of sequencing are obvious. When it comes to profiling of microRNA, if you're really just interested in the differential expression of microRNAs, we would still recommend doing arrays or going the array route. And that's for a number of reasons. Besides the cost, of course, that is involved, it's really the sample prep, the lengthy library prep that is involved with next-gen sequencing. There are a couple of amplification steps, so we feel that and of course it's not just us who feel that, but there are a lot of people in the field that think that, or that are worried that you're biasing your microRNA profile during this lengthy library prep stage. Whereas in the array, of course, you go take your sample pretty much directly and put it on your chip. So the workflow is pretty much identical: sample QC, library prep, then the sequencing, and then different types of data analysis depending on what the customer chooses in our case. The instrument that we use in this case, if you really just look at the microRNAs, we use the Illumina Genome Analyzer, GA2X. If you look at transcriptome sequencing or something like that, of course we use the HiSeq. This machine has a read length of 35 bases, and the number of reads is typically around up to 30 million per lane. And so in the table below here, what it shows is basically the total number of reads, roughly around 30 million assumed here, and then the number of samples that you barcode or multiplex per lane. So it's really up to you how many samples you want to squeeze onto one lane. But of course, obviously, the more samples you put on, the lower the number of reads per sample becomes. So typically, we recommend 3, maximum 6 samples per lane. If you go higher than that, then you might really get better data with an array. When you run 6 samples and get 5 million reads, that's typically what people consider to be equivalent to a microarray, the depth of data. If you are interested in discovery of novel microRNAs or mutations, things like that, then we would recommend, of course, a lower number of samples, maybe even 1 sample per lane, so that you can take full advantage of the read depth for this particular sample. This is just as a reference for you to look through later if you're interested in this. This is the data processing with our software. Basically, in the red marked area here, this is what we deliver as standard data. It's not just the raw reads, but we, of course, transform it to mappable reads. And then this is here showing the process of further mapping to other genomes. The species genome, of course, first we try to map against the mammalian microRNAs that are existing in miRBase. And what you end up with are 4 different groups. And they range from group 1 here with a known microRNA species that were reconfirmed pretty much with the sequencing results to all the way group 4 with potentially novel microRNAs. And to give you an idea about the numbers, when you start out with 30 million raw reads that the sequencers gives you, you're losing a big chunk of data right at the beginning when you filter out all kinds of junk. It's you pretty much end up roughly around with 20 million reads then that are mappable. And then if you take the mappable reads and then go further to the workflow as I just showed you, then you pretty much end up with this is of course just one case study here, but for group 8, so 8 million reads were found to be already existing microRNAs, and in group 4 there are more than 3 million reads still available for or found in this case for potentially novel microRNA transcripts. Just to give you an idea about the numbers. So in general, the data amount and the type of data that you get from sequencing is, of course, is unmatched compared to an array, and on the other hand, this oftentimes overwhelms first-time users. So one of the nice things that you're getting is also you get the length distribution of mappable reads. So microRNAs in mammalian are typically up to 22 nucleotides, and in plants longer. So this could be either a sample that is a plant sample, or it's a specific developmental stage, because the length distribution varies based on the developmental stage also in mammalian. And just one other point that I want to highlight in terms of the data that you're getting is, and there's still a lot of discussion about this, how much of this is really true and how much of this is a library prep artifact. It's the existence of so-called isoforms or isomers. So if we start out here at the very on the top row here, this is the microRNA that you would probably find in miRBase. So it has 60,000 reads for just this species of the microRNA. But then there's a second microRNA which is just shortened by 1 nucleotide, which still gets more than almost 11,000 reads. So that's quite a significant number still. And then, of course, you have different isoforms. Here, this one, the third one in the third row is shifted by 1, and you have 2,000 reads. And then you have a whole number of forms, isoforms that have a rather low number of reads. So yeah, this is something to keep in mind when you are looking at array data also versus validation experiments with qPCR, because of course the array won't be able to distinguish between these first three, for example, whereas a highly specific qPCR method, like let's say like you use a TechMan system, that will differentiate between these. So it's not a surprise that oftentimes the array data don't exactly match the qPCR data. We are in general happy if the direction of regulation is identical, and that's something that we would then consider validated. And to give you an idea about the percentages, typically when you have regular tissue samples, usually we see a validation rate of 80, 85, sometimes even 95% between array versus qPCR data. When you go to cell line samples, for some reason this is lower. It's typically around 70%. And looking at some papers that were published over the last 2 years in this field, there are of course the different fields like I mentioned. There's the diagnostic and the therapeutic field, and here are just some samples about using microRNAs as biomarkers. In this case here, it was a study using pancreatic cancer, There were 10 patient samples versus 10 control samples were analyzed, and the results that were found are that 37 microRNAs are downregulated and 54 are actually upregulated. And the interesting thing about this study was it was already a study. It was published in 2010, but it was already a study that used plasma samples. And the major findings between the up and down regulation, of course, was that the expression of miR21 was correlated with the worst survival rate, and on the other hand, LED7 was inversely correlated with the survival. And so basically, this is a typical example where the microRNA expression profiles can indicate, even in plasma, can indicate the state of the cancer and the aggressiveness of the cancer, which was determined based on the miR21 expression levels. Then another body fluid here as a case study, it's microRNA expression or leakage of expression into breast milk. In this case, the study compared microRNAs of MCF7 in both the cellular and the extracellular part, and was done using microarrays. And it was found that basically the two particular microRNAs were found increased in breast milk if cancer was present. And these microRNAs were microRNA 451 and 1246. So again, a very neat, in a non-invasive way of determining cancer and diagnosing cancer. It was basically using this selective occurrence of microRNAs in breast milk. In the healthy patients or controls group, these particular microRNAs did not show up in breast milk. Then one of my favorite papers, simply because it's visually really clear what's happening, is this paper, which is again in the molecular diagnostics field, and it's a study trying to differentiate between 4 different types of kidney cancer. So what you're seeing here is on the right is a normal group, and then you have 4 different subtypes of kidney cancer. And like with many cancer types, it's oftentimes difficult to almost impossible to diagnose them properly, and as a clinician, just by looking at them. And yeah, this experiment clearly shows that the microRNA expression pattern can be used to clearly diagnose and differentiate between these 4 different subtypes. In this case, a total of 94 patients were analyzed, and it was done the way that some samples were known, with known subtypes were included, as well as some with unknown, and then the diagnosis was later confirmed. This is a similar experiment was in the colorectal cancer field. It's one of the most common cancers in the Western world, and can be cured typically when diagnosed early or detected early enough. And this paper deals with the differential expression of miR150, which is decreased in the transition from normal to colorectal cancer, depending on the stage. So again, miR150, clear biomarker associated with the prognosis and outcome of the cancer. At this point, I would like to mention, typically, this is a rare case, and probably in the meantime, one might look different at this. So typically, as mentioned before, it's not just one particular microRNA that is involved and that you should look at. It's typically an entire network of, there are a couple of studies out there that suggest that the range of, that the minimum profile, signature profile that you should look at is anywhere between 15 to 20 microRNAs. When you go from, still in the molecular diagnostic field, but when you move to the environmental or chemical toxins, this is a study with Ritalin and Ritalin samples versus control samples. And again, 47 microRNAs were found to be significantly up or down regulated by Ritalin treatment. And there are two types of analysis shown here. Let's start out on the right here. It's just a regular t-test, a heat map with the Ritalin samples clearly showing a differential pattern versus the control samples. And then here shown on the right is a principal component analysis, and shows also extremely nice clustering of the red control samples versus the purple and blue Ritalin treated samples. And one of the, when we move on from the purely diagnostic field to the therapeutic field, there was a paper which is now already almost 4 years old, but it's to the best of my knowledge, it was the first time that microRNAs were used as a that there was a hint for a therapeutic function of microRNAs in the breast cancer field. And basically what was done here is not only differential profiling of the different breast cancer samples versus control samples. And of course what the authors achieved was that they saw the signature profile, which microRNAs were up and down regulated. But then they went the additional step and looked at what would happen if they reinstate expression of certain microRNAs. And highlighted are here miR335 and miR126, which upon reinstating their expression levels to precancer state, to precancer levels, they were shown to suppress metastasis. So that was a really very important paper, especially at the time. And this was done in vivo, so really exciting. This is a study where, again, which took it a step further and looked at reinstating what happens if you restore expression. In this case, it was microRNA 342, and it was dealing with the again, cancer samples, of course, and cancer samples that were resistant, showed chemical resistance to tamoxifen. And what is shown here on the top is that the cell growth of the tumor was really high for untreated samples, of course, and then these are different control samples. And then here on the very right, with the in dark red shown are the samples where the expression of miR342 was reinstated. And as you can see, cell growth dramatically goes down. And basically what this expression, reinstated expression of this particular microRNA did was that it allowed the chemotherapy to work again. And so you can see this here. As soon as you restore the microRNA expression, the apoptosis rate really shoots up. This as a brief overview of some of the latest publications in the field. And as mentioned, we're globally represented pretty much. If you're in a country that's not listed here, like I said, we have samples. We receive samples every day from many different countries, and especially, of course, from Europe and the United States. And yeah, all samples end up in Houston, so you can send them directly to us, which is the typical route for people in Europe, of course. And yeah, I hope this was somewhat helpful to you and give you at least a brief overview. As mentioned, we will send you the PDF file with all the links so that you can look at the particular papers if you're interested in them. And yeah, you will also receive the link to a recording of this webinar.